Hello and welcome. So today's topic is our contents of eyeball or the structures present inside the eyeball. Remember in exams, in Viva, so many examiners ask you that tell me the structures present inside the eyeball or the contents inside the eyeball, and you start saying that iris, sclera, cornea, then ciliary body, choroid, lens, aqueous humor, vitreous humor. No. Try to understand these conjunctiva, cornea, sclera, these are the parts of the outer layer of the eyeball. Iris, ciliary body, choroid, these are the parts of middle layer of the eyeball. Retina is the single most entity, okay? It has no parts. Now, there is only three structures present inside the eyeball, or the eyeball has three contents inside. First of all, aqueous humor, then vitreous humor and lens. Only these three structures. So, first and foremost, our aqueous humor, as you can see in this photo. As I told you in my earlier videos that our entire eyeball has two segments. One is anterior segment, posterior segment. Behind the lens, posterior segment. In front of the lens, anterior segment. Anterior segment has two chambers, anterior chamber, posterior chamber. Both chamber has aqueous humor, okay? In anterior chamber, it is almost 0.25 ml, and in posterior chamber, it is almost 0.06 ml. Posterior chamber is very less because the space in the posterior chamber is very less. The function of this aqueous humor is basically it provides the nutrition, then it maintains the intraocular pressure or IOP. What is IOP or intraocular pressure we'll talk about later in clinical chapter, not here. It maintains the optical transparency along with the, it clears the remnant or the debris like macrophages, then phagocytes, any effective foreign bodies, etc. Now the refractive index of this aqueous humor is almost 1.336, and the components of this aqueous humor is 99.9% of water along with the 0.1% of solids. These are protein or in amino acid, which are basically forms in colloid matter or colloid forms, glucose, urea, ascorbate, lactic acid, inositol, sodium, chloride, calcium, magnesium. How it forms, what are the local factors, these things I will talk about in physiology chapter, not here. Now the vitreous humor, it is inert jelly-like substance. It is also crystal clear, transparent, and in anterior part, as you can see in this photo, in an anterior part of this vitreous humor, it has cup-shaped depression area for the placement of the lens. It is almost 4 milliliter in amount and 4 gram in weight, and basically stabilizes the posterior part of the eyeball and nourishes the posterior part of the lens and the entire retina along with the optic disc. Okay, now the composition of this vitreous humor is basically it is consists of 99% of water along with the randomly interspersed collagen fibrils along with the hyaluronic acid, then hyalurocytes and specialized mucopolysaccharides. Okay, now try to understand this vitreous body has two parts. One is cortex and medulla. First of all, cortex. Cortex means basically the peripheral part of the vitreous, which is posteriorly bounded by the retina and anteriorly bounded by the lens and the ciliary body and the zonules of Zinn. What are these things we'll talk about later. And I already told about this ciliary body in my earlier videos, okay? And the medulla or the main body of the vitreous is just jelly-like structure. It is just formed by the collagen and the hyalurocytes and the hyaluronic acid and the mucopolysaccharides. That's it. Now, one thing as you can see in this photo that in the center of this vitreous body from the optic disc of the retina to the posterior part of the lens, there is a canal or a space. This space is known as hyaloid canal. It is also known as Clockett's canal. Okay, it is anteriorly attaches with the posterior surface of the lens via ligaments of Weigart or Wigert. You can say whatever you want. And posteriorly or in the periphery, it is attaches with the internal limiting membrane of the retina and around the margin of the fovea and around the margin of the optic disc. That's it, as you can see in this photo. How this vitreous body forms, what are the mechanism of function, these things you have to know about in physiology chapter, not here. And now the last point or the last topic of today's video is the lens. As you can see in this photo, this lens is actually biconvex structure. Okay, biconvex structure, crystal clear, transparent, and it presents in between the iris and the vitreous body. In the vitreous body, I just told you now that anterior surface of the vitreous body, it has cup-shaped depression area for holding or the attachment of the lens, which is known as patellar surface of the lens. Anyway, so the lens is almost 9 to 10 millimeter in diameter. Now the attachment of the lens, as I told you just now, that this lens is attached with the vitreous body through its ligament of Wigert or Wigert. It is also known as retrolental ligament. I mean the ligament present in the posterior surface of the lens. And in the periphery, it is attached with the entire ciliary body, whole ciliary body, through its zonules of zinc. It is covered by a thin, transparent hyaline capsule or the hyaline membrane. Then you're going to find epithelium of the lens, which is present only in the anterior surface, not in the posterior surface of the lens. Now, in the anterior surface, this epithelium presents at the equator or at the center. It is columnar, and at the periphery, it is cuboidal. Nucleus of the lens, which is multi-layered. First of all, you're going to find the embryonic nucleus or the embryonic part. Then you're going to find fetal part. Then you're going to find infantile part, and then adult part, and lastly the cortex. Okay, so this is all about the structures or the contents of the eyeball, including our aqueous humor, vitreous humor, and the lens. See you soon. Till then, bye.